How does the oxygen we breathe keep us alive? What is the oxygen used for? In aerobic metabolism, molecular oxygen accepts the electrons that originate from energy-rich carbon compounds, such as the foods we eat. Because oxygen has a high affinity for electrons relative to carbon, a lot of energy is released when it accepts the electrons. The hemoglobin in blood carries molecular oxygen to our brain, heart, and other tissues that require a constant source of energy. Without oxygen, it's not possible for us to meet our energy needs, and the cells in these tissues start to die. This is one of the reasons that severe blood loss causes extensive tissue damage, including brain damage. Blood not only transports oxygen, but also the other raw materials such as glucose that we need to stay alive. In this animated presentation, we'll learn how the transfer of electrons from glucose to molecular oxygen generates ATP. Energy is released when electrons are transferred from energy-rich forms of carbon to molecular oxygen. Electrons are represented with the small letter E and a minus sign. How is this energy used to make ATP? Inside the cell, specialized protein complexes couple the flow of electrons to the transport of protons out of the mitochondrial matrix. When these protons spontaneously flow back in, they drive a tiny molecular motor that synthesizes ATP. This model for ATP synthesis is called chemiosmosis. Chemiosmosis is the coupling of ATP synthesis to electron transport through the formation of a proton gradient. As we learned in the last chapter, the reduced coenzyme NADH links glycolysis and the TCA cycle to ATP synthesis. NADH transfers electrons to a protein complex in the inner mitochondrial membrane. The electrons flow through several complexes and eventually reach molecular oxygen. Oxygen is the final electron acceptor in the electron transport chain. As the electrons flow through the protein complexes, protons are pumped out of the mitochondrial matrix. When the protons flow back in, ATP is synthesized. This process is called oxidative phosphorylation. Oxidative phosphorylation is the phosphorylation of ADP coupled to the transfer of electrons to molecular oxygen. In this activity, we will explore the process of oxidative phosphorylation. First, we'll learn about the electron transport chain located in the inner mitochondrial membrane. Then, we'll see how the free energy released by the transport of electrons is used to drive the synthesis of ATP. We'll finish by looking at experimental evidence that supports the model of chemiosmosis. Electron transport and ATP synthesis take place in the cell's mitochondria. Recall that mitochondria have two membranes, an inner membrane and an outer membrane. The outer membrane has channels that allow the passage of small molecules. The outer membrane is separated from the inner membrane by the intermembrane space. The inner membrane encloses the mitochondrial matrix. This membrane is impermeable to small molecules and ions. As we'll see, specific proteins are needed for transporting molecules and ions across the inner mitochondrial membrane. The electron transport chain is made up of a series of protein complexes located in the inner mitochondrial membrane. The complexes pass electrons from molecules like NADH to molecular oxygen. The electrons are transferred spontaneously when they move from molecules with low electron affinity to molecules with higher electron affinity. The electron affinity of a molecule is measured by its reduction potential, its tendency to gain electrons. Reduction potential is symbolized by capital E. Several different types of molecules are found in the electron transport chain. Flavoproteins, such as FMN and FAD, can accept 1 or 2 electrons. They make excellent bridges between 2 electron donors, such as NADH, and 1 electron acceptors. Coenzyme Q also participates in 1 or 2 electron transfers. Examples of 1 electron acceptors are iron-sulfur proteins, copper-containing proteins, and cytochromes. Like hemoglobin and catalase, cytochromes contain a heme group. Heme groups have iron centers. The heme iron is converted from iron plus 2 to iron plus 3. Iron plus 2 has 1 more electron than iron plus 3. Recall that the TCA cycle takes place in the mitochondrial matrix. The coenzymes FAD and NAD are reduced as acetyl-CoA cycles through. The reduced coenzyme NADH transfers 2 electrons to the FMN flavoprotein, the electron acceptor, on complex 1. The electrons flow from FMN through an iron-sulfur center and are passed on to coenzyme Q. In the TCA cycle, the enzyme succinate dehydrogenase, also known as complex 2, was tightly bound to the mitochondrial inner membrane. Its coenzyme FAD accepts 2 electrons from succinate. The succinate electrons also get passed on to coenzyme Q. Coenzyme Q is a hydrophobic molecule that's membrane-soluble. Coenzyme Q shuttles electrons from complexes 1 and 2 to complex 3. Complex 3 passes the electrons from coenzyme Q to cytochrome C. Unlike coenzyme Q, which is hydrophobic and membrane-soluble, cytochrome C is hydrophilic and water-soluble. Cytochrome C shuttles electrons to complex 4. Complex 4 is the last complex in the electron transport chain. In complex 4, electrons are transferred through heme and copper centers to molecular oxygen. The transfer of electrons to molecular oxygen completes the flow of electrons through the transport chain. FAD is second, coenzyme Q is third, and cytochrome C goes last. Cytochrome C has the highest potential to accept electrons. It's positioned closest to molecular oxygen in the electron transport chain. The spontaneous flow of electrons through the transport chain is coupled to the movement of protons out of the mitochondrial matrix. The protons are moved from a region of low concentration to a region of high concentration, and this requires the input of energy. Whereas some reactions are driven by the hydrolysis of ATP, the movement of protons out of the matrix is driven by electron transport. As the 2 electrons from NADH are transferred through complex 1, 4 protons are moved out of the matrix. 4 more protons are transported out of the matrix as 2 electrons are transferred from coenzyme Q to cytochrome C through complex 3. This mechanism is known as the Q cycle. The electrons are transferred from coenzyme Q to cytochrome C molecules. Cytochrome C delivers the electrons to complex 4. More protons are pumped out of the matrix as electrons move through complex 4. 1 proton is pumped out for every electron that enters complex 4. The ultimate electron acceptor is molecular oxygen. It takes 4 electrons and therefore 4 cytochrome C molecules to reduce 1 oxygen molecule to 2 water molecules. The electron transport chain can be disrupted. When this happens, cellular respiration stops. The compound cyanide is a well-known inhibitor of electron transport. It disrupts complex 4 and prevents electrons from reaching molecular oxygen. Cyanide poisoning is a little bit like locking the gears of a machine by inserting a metal rod. Smoking cigarettes and breathing smoke-filled air during fires are major sources of cyanide exposure. Because electron carriers cycle between oxidized and reduced forms, the blocking of electron transfer to molecular oxygen disrupts the entire transport chain. If the flow of electrons is disrupted, ATP can't be made, and aerobic organisms which depend on oxygen die. Cellular respiration depends on the flow of electrons to molecular oxygen. In the absence of such inhibitors, the flow of electrons is coupled to the transport of protons out of the matrix. The removal of protons from the mitochondrial matrix does two things. It creates a concentration gradient across the inner membrane as the concentration of protons outside the matrix exceeds the concentration inside. It also builds up an electrical potential because a positive charge is removed every time a proton is transferred out of the matrix. The combination of proton concentration and electrical gradients, called an electrochemical gradient, creates the proton motive force. The proton motive force is the energy stored in a proton gradient. When this energy is released, ATP is synthesized. How does the proton motive force make ATP? As you will see, there's another protein complex, complex 5, also known as ATP synthase. ATP synthase is a rotating molecular motor that contains a proton channel. When protons flow through this channel, ATP is synthesized. ATP synthase is made up of two distinct components. One component is in the membrane. It has a ring of alpha helices through which the protons flow. The other component contains the enzyme's 3 active sites. Protons flowing through the first component drive conformational changes in the second component. ADP and phosphate bind to one of the active sites. Conformational changes drive the ADP and phosphate together to form ATP and then force it out of the active site. In 1997, the scientists Paul Boyer and John Walker were awarded the Nobel Prize for their work on how the ATP synthase enzyme works. The newly formed ATP is transported out of the mitochondrial matrix. Its export is coupled to the import of ADP. ATP carries a greater negative charge than ADP, so its export is favored by the membrane's electrical potential. Like ADP, phosphate must also be transported into the matrix. The phosphate carries a negative charge. Because phosphate carries a negative charge, its transport into the matrix is not spontaneous. Its movement into the matrix is coupled to the return of a proton. Now that we've seen how the proton motive force is used to synthesize ATP, let's learn how some of these details were discovered. When scientists discovered that organisms oxidized food for energy, They wondered how the oxidation energy was captured. Researchers eventually determined that ATP and electron transport were involved. Scientists proposed various models to explain how these two processes were linked. In 1961, the biochemist Peter Mitchell proposed the chemiosmotic model, which is the model we have just presented. Mitchell proposed that ATP synthesis is coupled to electron transport by the formation of a proton gradient. Experimental results support the chemiosmotic model, and Mitchell was awarded a Nobel Prize for his work in 1978. Let's see how the chemiosmotic model is supported by experiments that disrupt the proton gradient. Inhibitors are valuable research tools. These molecules are often used to study enzymes and have important applications in the development of new drugs. One class of inhibitors disrupts the coupling of electron transport to ATP synthesis. They're called uncouplers. Unlike inhibitors that bind to specific enzymes, uncouplers are hydrophobic molecules that carry protons across the inner mitochondrial membrane. Uncouplers allow protons to bypass ATP synthase and re-enter the mitochondrial matrix. The energy stored in the proton gradient is lost as heat rather than used to synthesize ATP. Uncouplers destroy the proton gradient and prevent ATP synthesis. Would you expect an uncoupler to affect electron transport? Click in the box next to the correct answer, then click Submit. Yes, that's correct. Electron transfer reactions are spontaneous. The released energy is used to pump protons out of the matrix. The ability of the protons to flow back into the matrix without entering complex 5 has no effect on the transfer reactions. ATP synthesis is not spontaneous. It requires the energy released as the protons flow back into the matrix. Some uncouplers are made by an organism to regulate its metabolism. Uncouplers create a membrane channel through which protons can flow. Newborn babies have high levels of the uncoupler thermogenin to help them keep warm. Leptin is a small protein that helps regulate body weight. It's secreted by fat cells. Some people and animals that are obese have abnormal levels of leptin in their blood, or defective leptin receptors on their cells. In healthy animals, leptin levels rise when food calories exceed their needs. This does two things. Appetite is suppressed, and the levels of an uncoupler increase. The uncoupler speeds up the organism's metabolism, and more calories are lost as heat. Two Nobel Prizes were awarded for the research described in this activity because of the importance of the work to our understanding of cellular respiration and metabolism. The energy released when food molecules lose electrons to molecular oxygen is converted into the potential energy of a proton gradient. The energy conversion takes place in the inner mitochondrial membrane. Specific protein complexes in the membrane couple the transfer of electrons to the movement of protons out of the mitochondrial matrix. ATP is made when the protons are allowed back into the matrix through complex 5, which is ATP synthase. The protons turn a molecular motor that drives conformational changes in the ATP synthase enzyme. The conformational changes drive the formation of ATP from ADP and phosphate. The synthesis of ATP is a highly regulated process. Uncouplers are used to disrupt the proton gradient, thereby uncoupling electron transport from ATP synthesis. This alters an organism's metabolic efficiency and is important in the regulation of body temperature and body mass. We've now seen how aerobic respiration works. Because molecular oxygen has a strong affinity for electrons, a lot of energy is released when it accepts them. The released energy is first transformed into a proton gradient. When the protons flow back into the mitochondrial matrix, the released energy turns a tiny molecular motor that synthesizes ATP. The ATP is then used to power other processes within the cell.